Okay, so picture this. It's the 1780s, right? And this Italian scientist, Lazzaro Spallanzani, he intentionally swallows these little linen bags.
Right, and perforated wooden tubes, too, filled with chunks of meat.
Yeah, wooden tubes.
Mm.
And he just, waits a while, intentionally vomits them back up, and then actually squeezes the fluids from inside those tubes into glass vials on his desk.
It sounds completely unhinged.
Totally. It sounds like madness. But he was actually trying to solve this, like, profound mystery that happens inside you every single time you eat lunch.
Yeah, I mean, it is arguably the most profound transition in biology.
Right. We all grab a turkey sandwich, take a bite, and give absolutely no thought to the wild, almost magical mechanism where that sandwich, stops being food and literally becomes you.
Exactly. We're taking the outside world, bringing it inside, and completely dismantling it on a molecular level just to build our own cells. And for the longest time, humanity really had no idea how we pulled that off.
See, I always assumed the historical consensus was just that the stomach acted like a giant, highly acidic blender, like a swirling vat of physical mush.
A lot of people still think that, actually.
Right. But reading through the research we pulled for this deep dive, the modern reality of how we handle carbohydrates, proteins, and fats is just staggeringly precise.
It's incredible.
It's way less of a blender and more of a highly choreographed chemical deconstruction site. So our mission today is to map that incredible journey, debunk some massive gut misconceptions, and figure out what actually happens to that sandwich.
I love it. Let's do it.
Okay, let's unpack this. The modern medical term for this process is chemical digestion. But what are we actually talking about on like a microscopic level?
On a microscopic level, we are talking about enzymatic hydrolysis.
Enzymatic hydrolysis.
Right. So we consume these large, structurally complex macromolecules, polysaccharides, which are complex carbs, dietary proteins, and triglycerides, which are fats.
And they're huge, right? Like
Right.
physically too massive to just pass through the intestinal wall.
Exactly. The entire goal of our digestive tract is to break them down into singular units that are small enough to actually cross the intestinal epithelium and enter the bloodstream or the lymphatic system.
Hydrolysis is the operative word there. It sounds like we're just dissolving things in water, but the chemistry is a lot more targeted than that, isn't it?
Oh, very targeted, yeah. An enzyme doesn't just melt your food, it acts more like a highly specific pair of chemical scissors.
Chemical scissors, I like that.
Yeah, so the enzyme finds a very specific chemical bond, say a peptide bond, that's holding two amino acids together in a protein, and it perfectly positions a single water molecule right across that bond.
Oh, wow.
And it uses the water to physically snap the link. It does this millions of times, bond by bond. So ultimately, your long-chain carbs end up as simple monosaccharides, like glucose. Proteins are reduced to free amino acids, and fats are cleaved into free fatty acids.
That makes the body sound like an incredibly efficient, sterile laboratory.
Yeah.
But going back to Spallanzani and his wooden tubes, I mean, we didn't always view the body as a chemical lab.
Not at all.
The historical debate over this was shockingly aggressive from what I read.
Oh, it completely dominated early medicine. Throughout the 17th and 18th centuries, physiologists were really split into two warring camps over what digestion actually was.
The iatromechanists and the iatrochemists, right?
Right, so you had the iatromechanists, who were mechanist physicians. They observed that birds had gizzards filled with stones, and they concluded that human digestion must be trituration.
Wait, so they genuinely believed the human stomach was just like a muscular millstone mechanically crushing food into paste?
That was the prevailing theory, yeah. And then on the opposing side, you had the iatrochemists. They argued that digestion was entirely chemical, specifically like a form of fermentation or even putrefaction.
Putrefaction, like rotting.
Yeah, like the food essentially rotted in a controlled way inside you. The frustration was that both camps had elements of the truth, but they just lacked the experimental tools to settle the dispute.
Which brings us to the bizarre lengths they went to for an answer. I mean, Spallanzani swallowing wooden tubes is wild, but my absolute favorite has to be the French physicist from 1752, René-Antoine Fréchot de Réaumur.
Oh, yeah. The kite experiment?
Right. He didn't use himself. He used his pet kite, which is a bird, right?
A rather large bird of prey, yeah.
Yeah, raptor. So Réaumur drills holes in these tiny metal tubes, packs them with chunks of meat, and slips sponges inside. He feeds them to the bird, knowing raptors naturally regurgitate indigestible bone and metal.
Which is pretty clever.
Super clever. And when the bird threw the tubes back up, he found the meat inside was partially dissolved, even though the metal tube protected it from any physical grinding. and the sponges were totally soaked with this mysterious fluid.
He had managed to extract pure gastric juice.
Exactly. He squeezed that fluid out of the sponges, applied it to a fresh piece of meat sitting on a dish outside the bird's body, and just watched it dissolve. It was undeniable proof that gastric juice was a chemical solvent, not just some biological lubricant for a grinding stomach.
And that concept completely laid the groundwork for William Beaumont in the 1820s.
Oh, the guy with the fur trader.
Right. Beaumont was an American army surgeon who treated a fur trader named Alexis St. Martin. St. Martin had suffered this devastating gunshot wound to the abdomen at very close range.
Yikes.
The wound miraculously healed, but it left a permanent gastric fistula.
Which is basically an open literal window straight into the man's stomach, which sounds horrifying.
It is, but Beaumont essentially turned the poor guy into a living laboratory for a decade. He would tie pieces of food to silk strings, physically drop them into St. Martin's stomach through the hole, pull them out hours later, and meticulously log the digestion times.
That is dedication. Gross, but dedication.
Very. He found that boiled rice broke down in about an hour, whereas raw cabbage took significantly longer. He was also the one who definitively confirmed the presence of highly concentrated free acid in the stomach.
So what does this all mean for the big picture? Like, why did it matter so much that digestion could happen in a test tube or outside the body?
Well, if we connect this to the bigger picture, these specific, gritty experiments were the death blow to a lingering medical philosophy called vitalism.
Vitalism.
Right. Vitalism was the almost mystical belief that biological processes required a special living force that was unique to a living body.
Oh, so like biological magic.
Exactly. By proving that you could extract gastric juice, put it in a glass vial on a desk, and achieve the exact same chemical breakdown of a piece of meat, they proved digestion didn't require magic. It demonstrated that human beings are spectacular, mechanistic chemical machines.
Okay, so knowing we are chemical machines, let's look at how that machinery handles the basic carbohydrate. If I take a bite of a piece of bread, I've always assumed the stomach was the main event. But the biochemistry actually kicks off the second the food hits your saliva, right?
It initiates immediately in the mouth. Your saliva contains an enzyme called salivary amylase. Historically, it was known as ptyalin.
Right.
Its singular job is to hunt for alpha-1-gal-4 glycosidic bonds. Those are the specific links holding together long chains of glucose in starch.
Gotcha.
Salivary amylase starts cleaving those massive chains into shorter fragments called dextrins and a double sugar called maltose while you are still chewing.
But the research noted a massive interruption here. I had this visual of salivary amylase like riding down into the stomach with the bread and just continuing to chop up carbs. But the stomach acid actually violently shuts the whole thing down.
Yeah, the environment changes way too drastically. Human stomach acid sits at a pH of about 1.5 to 2. It is incredibly caustic.
Basically battery acid.
Pretty close. And enzymes are highly sensitive, three-dimensional proteins. When salivary amylase hits that wall of acid, it denatures. Its shape physically unwinds and collapses.
So it's basically an assembly line that gets abruptly halted because the conveyor belt passes through a blast furnace. The workers are completely incapacitated.
That's a great way to put it. Carbohydrate digestion just comes to a screeching halt in the stomach. It goes completely dormant. The carbohydrates sit there untouched while the stomach focuses on other things. It's only when that mixture, which is now a highly acidic, semi-liquid paste called chyme, exits the stomach and enters the small intestine that the process frantically resumes.
And how does it start back up?
The pancreas secretes an alkaline bicarbonate fluid to neutralize the stomach acid alongside a fresh batch of pancreatic amylase. And that picks up exactly where the mouth left off.
And it all culminates at the brush border. Let's define brush border real quick because it's a vital piece of intestinal anatomy. We were talking about the microvilli.
Right. Yes, the microvilli are these microscopic hair-like projections that densely line the inner wall of the small intestine. They look like the bristles of a brush under a microscope, hence the name.
Right.
They increase the surface area of the gut exponentially. And embedded directly into those bristles are the final, highly specialized enzymes, maltase, sucrase, and lactase.
So they act as the absolute finish line. They take those small double sugar molecules and snip them one last time into single monosaccharides, glucose, fructose, and galactose.
Exactly.
And this is where specific transporters come into play to get them into the blood.
The precision of those transporters is remarkable, actually. Glucose and galactose are actively pulled across the intestinal wall by a transporter called SGLT1, which uses a sodium gradient for power.
And fructose.
Fructose, on the other hand, slips through a completely different door. a passive transporter called GLUT5.
Which perfectly explains why we can't digest fiber. Like a piece of celery and a potato are both technically carbohydrates. They are both chains of glucose. But earlier you mentioned salivary amylase only targets alpha-1 Gul-4 bonds. Fiber is structurally different, isn't it?
It is. Fiber is made of cellulose. The glucose units in cellulose are locked together by beta-1,1,4 bonds. It is a very slight geometric difference in how the molecules connect, but human amylase simply cannot read that shape.
Wow.
We just do not possess the biological scissors to cut a beta bond.
So the celery passes through the stomach, and the small intestine completely invisible to our enzymes.
Completely untouched by human biology. We rely entirely on the trillions of bacteria living in our large intestine, the colon, to deal with it.
Because they have the tools we don't.
Right. Those bacteria have the enzymes we lack. They ferment the fiber, breaking it down and releasing short-chain fatty acids like butyrate as a byproduct. So any caloric energy you derive from dietary fiber isn't from your own digestion.
It's one of the bugs.
Exactly. It's a metabolic kickback from your bacterial colonies. Modern nutrition science reflects this by drawing a very hard line between digestible carbohydrate and fermentable fiber.
That geometric specificity with enzymes also clarifies lactase persistence, I think. Lactase is the brush border enzyme that splits lactose, the sugar found in milk. And I think a lot of people operate under the misconception that if you are lactose intolerant, your body rejects the entirety of the dairy product.
That is a very common misunderstanding. Lactose intolerance has nothing to do with dairy fat or dairy protein. Your body digests the lipids and whey perfectly fine.
Really? Just the sugar is the issue.
Just the sugar. It is exclusively the failure to break the chemical bond of the milk sugar. And historically speaking, losing that ability is the default human condition. Roughly 65% of the global adult population naturally stops producing sufficient lactase after they are weaned from breast milk.
So the ability to drink a glass of milk as an adult is actually a genetic mutation.
A relatively recent evolutionary trait, yes. Lactase persistence only emerged within the last 7,500 to 10,000 years, strongly tied to populations that adopted dairy pastoralism.
Okay, so if the stomach is basically a hostile wasteland that actively destroys carbohydrate enzymes, why do we even have a vat of KH 1.5 acid sitting in the middle of our body? It seems wildly counterproductive.
It would be if carbohydrates were the only thing we ate. Aside from sterilizing our food and killing ingested microbes, that highly acidic environment is the specific trigger required for the next massive biochemical hurdle, dietary protein.
And digesting protein is inherently dangerous, right? I mean, our organs, the stomach wall, the intestines, the pancreas, they're all built out of protein.
Exactly.
If our cells just secreted active protein-digesting enzymes, those enzymes wouldn't know the difference between a dietary steak and the stomach lining. They would literally dissolve our own organs.
The resulting tissue damage would be catastrophic. To prevent self-digestion, the body utilizes this brilliant activation cascade. These specialized chief cells in your stomach don't release an active enzyme. They secrete a completely harmless, inactive precursor called pepsinogen.
So it's shipped inactive. It almost sounds like ordering a set of incredibly sharp kitchen knives. You would never ship them loose in a box where they could cut through the cardboard. You ship them locked inside thick plastic sheaths. And the stomach acid is what finally pulls the sheaths off.
That is a perfect analogy. When that harmless pepsinogen makes contact with the extreme hydrochloric acid in the stomach lumen, the acid chemically alters its shape.
It pulls the sheath off.
Right. It unpins the grenade, so to speak, converting it into active pepsin. Pepsin is an endopeptidase. It immediately starts attacking the internal bonds of dietary proteins, chopping long polypeptide chains into smaller pieces. And it only does this safely within the protective mucus lining of the stomach.
But the stomach eventually empties into the small intestine where there is no acid. How does the pancreas safely deliver its own protein enzymes without the acid trigger?
The pancreas uses a different master switch. It pumps out a whole family of sheathed inactive precursors: trypsinogen, chymotrypsinogen, and procarboxypeptidase.
That's a mouthful.
Yeah. But they float harmlessly into the small intestine. Waiting for them on the brush border is a single, highly specialized enzyme called enteropeptidase.
So enteropeptidase acts as the trigger.
Precisely. Enteropeptidase clips the protective sheath off just a tiny amount of trypsinogen, turning it into active trypsin. Trypsin then acts as an autocatalyst.
Meaning it activates itself.
It rapidly activates the rest of the trypsinogen, yeah, and then moves on to unsheath all the other pancreatic precursors. It creates this localized, self-amplifying wave of protein-cutting power exactly when and where the chyme is present.
Wow. And then the brush border peptidases finish the job, reducing these chains down to free amino acids, or tiny 2 to 3 amino acid chains, which are pulled into the blood by dedicated transporters like peptide-1.
Right.
Speaking of amino acids, there's a massive dietary myth we need to address here. In 1971, Frances Moore Lappé published a wildly influential book called Diet for a Small Planet. She argued that because plant proteins are incomplete, meaning they lack sufficient levels of certain essential amino acids, vegetarians had to meticulously combine complementary proteins like beans and rice in the exact same meal.
Yeah, that logic was based on a fundamental misunderstanding of the digestive timeline. Lappé's premise relied on a real physiological fact, which is that plant proteins generally do have limiting amino acids, but the dietary extrapolation that you must balance them on a single plate was completely unnecessary.
Because the body isn't processing meals in strict isolation.
Exactly. Physiological research clarified that the liver and the bloodstream maintain a circulating free pool of amino acids throughout the day. You can eat a bowl of rice at noon and a serving of beans at dinner.
And you're fine.
Perfectly fine. Your body absorbs the amino acids from both distinct meals, pools them together, and seamlessly synthesizes complete proteins. Lappé herself commendably walked back the strict combining rule in later editions of her book, but the myth remains incredibly pervasive.
It's fascinating how a slight misunderstanding of gastrointestinal transit times can shape the dietary anxiety of a whole generation.
Wow, absolutely.
Well, we've conquered carbs and proteins, which both dissolve relatively easily in the watery fluid environment of the gut.
Yeah.
But fats are entirely hydrophobic. They actively repel water. What happens when dietary oil hits a water-based digestive tract?
It creates a massive mechanical challenge. If you pour olive oil into a glass of water, it clumps together into a single impenetrable layer.
Right, it just floats on top.
Right. In the gut, enzymes can only work on the surface of a fat globule. They can't penetrate the interior. So if the fat stays clumped together, digestion would take infinitely too long.
Here's where it gets really interesting, because I always heard that bile digests fat. But looking closely at the biochemistry, bile doesn't actually digest anything, does it?
It does not. Bile contains absolutely zero digestive enzymes. It cannot break an ester bond.
Wait, so if bile isn't an enzyme breaking chemical bonds, what is its actual mechanical purpose in the gut?
Bile is a biological detergent. It is produced by the liver, stored in the gallbladder, and released into the small intestine when a hormone called CCK detects fat in the chyme.
A detergent, like dish soap.
Much like dish soap breaking up grease in a frying pan, yeah. Biomolecules have one side that loves water and one side that loves fat. They wedge themselves into those massive fat globules and physically tear them apart into millions of microscopic droplets. This process is called emulsification.
And I'm guessing that vastly increases the surface area so the actual enzymes have room to work?
That is where pancreatic lipase steps in. Aided by a smaller protein called collapase, which acts as an anchor to keep the enzyme attached to the slippery fat droplet, lipase hydrolyzes the triglycerides into free fatty acids and 2 monoglycerides.
Okay, so these broken down fat components are ferried to the intestinal wall by tiny vehicles called micelles. But this is the stage of fat digestion that blew my mind. Carbohydrates and proteins get broken down, pass through the intestinal cell, and go straight into the bloodstream. Fats get broken down, enter the intestinal cell, and then the cell immediately rebuilds them back into fat molecules.
Yeah, it's pretty counterintuitive.
Right. Why go through the immense biochemical effort of tearing a fat apart just to rebuild it inside the intestinal wall?
Because the rebuilt fats are then packaged along with cholesterol into these massive protein-coated cargo ships called chylomicrons.
Yeah.
The body builds them to safely transport hydrophobic fat through a watery body.
Okay, that makes sense.
But there is a structural problem. Chylomicrons are physically immense. They are far too large to fit through the tight junctions of the capillaries that feed into the bloodstream.
So they can't take the normal highway out of the gut.
No, they require a secret bypass route. Instead of entering the blood, these massive chylomicrons are extruded into lacteals, which are the specialized vessels of the lymphatic system located inside the intestinal villi.
So they go into the lymph nodes.
Into the lymphatic system, yeah. They travel slowly up through the lymphatic system, completely bypassing the liver's first pass filtration, and are eventually dumped directly into the venous bloodstream near the neck via the thoracic duct.
Wait, so the fat from my turkey sandwich travels up a lymphatic highway in my chest and drops into my blood near my neck.
It does, and the sheer volume of this transport is observable. If you were to draw a blood sample from someone shortly after they consumed a heavily fatty meal, the blood plasma wouldn't look clear. It would look visibly milky.
Oh, wow. The clinical term for that is lipemic, right?
Yes. It is a literal physical demonstration of millions of these massive chylomicrons circulating in the blood before the tissues have a chance to clear them.
We are talking chemical factories. The sheer scale of the fluid dynamics is staggering. The sources noted that every single day, the pancreas secretes about 1.5 liters of enzyme-rich juice, and the liver pumps out up to a liter of bile.
It's a lot of fluid.
All of it dumping into just a few centimeters of the duodenum, perfectly timed by hormones. So bringing this all together, why does understanding this exact chemistry matter for the average person or for modern medicine?
It matters deeply when we try to medically intervene or when a patient's anatomy changes. Take a common weight loss pharmaceutical like orlistat, sometimes sold over the counter as Ali. Its entire mechanism of action relies on the specific biochemistry we just covered.
How so?
Orlistat is designed to directly bind to and inhibit pancreatic lipase.
The specific enzyme that cuts the fat bonds. So if it's inhibited, the fat remains as intact triglycerides.
And because intact triglycerides cannot be absorbed by the brush border, they pass entirely through the gastrointestinal tract. This leads to a side effect known as steatorrhea, or heavily oily stool.
Unpleasant.
Very unpleasant. But it is a highly tangible proof of what happens when you subtract just one specific enzyme from that choreographed sequence.
It also explains conditions like cystic fibrosis, where patients suffer from pancreatic insufficiency. Their thick mucus blocks the pancreatic ducts, meaning the amylase, proteases, and lipases never reach the small intestine. They literally have to swallow capsules containing those exact enzymes with every meal just to avoid malnutrition.
Exactly. Or even celiac disease, which is fundamentally a failure downstream of protein digestion.
How does that work?
Celiac is a fascinating example. Certain peptide fragments found in gluten are uniquely resistant to being completely hydrolyzed by human proteases. In genetically susceptible individuals, those lingering intact peptide fragments manage to cross the intestinal barrier and are flagged by the immune system, triggering a severe autoimmune attack on the microvilli themselves.
Oh, that makes so much sense. I think this also brings a lot of clarity to bariatric surgery. In a gastric bypass, surgeons physically reroute the digestive tract, so food bypasses parts of the stomach and the duodenum entirely. You are fundamentally changing where and how fast food hits the small intestine.
You are.
So the dumping syndrome, those patients sometimes experience that intense surge of rapidly absorbed sugar and the brutal blood sugar crash that follows. That's basically those SGLT1 and GLUT5 transports getting completely ambushed because the stomach's metering system was removed.
Bingo. You are overwhelming the transport mechanisms. What's fascinating here is how these precise physiological truths also dictate our broader dietary boundaries, like the ongoing debate surrounding ketogenic diet.
The keto debate.
Right. Regardless of where one stands on the long-term efficacy of keto, there is a fundamental biochemical reality anchoring it. Human beings require essential amino acids, which we must obtain from dietary protein. We require essential fatty acids, which we must obtain from dietary fat. Our cells cannot synthesize them from scratch, but there is absolutely no such thing as an essential carbohydrate.
Because the body isn't helpless if carb digestion yields zero glucose. The liver can just manufacture its own glucose from non-carbohydrate precursors through a pathway called gluconeogenesis.
Correct. The brain and red blood cells demand glucose, but the body has a built-in load-bearing mechanism to synthesize it internally. That specific detail of our metabolic machinery is what makes completely eliminating dietary carbohydrates biologically survivable in the first place.
It all connects back to the cellular machinery.
Yeah.
So to recap the journey of that sandwich, digestion isn't some blunt force mechanical grinding. It is incredibly precise chemical hydrolysis. It's carbohydrates getting chopped by amylase, abruptly halted by stomach acid, and finished at the brush border. It's dangerous protein-cutting enzymes being shipped in sheaths and activated by an acidic alarm clock. And its fats being emulsified by bile detergents before taking a completely separate lymphatic highway to the neck.
It is an absolute marvel of evolutionary engineering. But before we wrap up, I want to leave you with one final, slightly mind-bending physiological fact about digestion that we haven't touched on yet.
Well, please do.
We have spent this entire time talking about what happens after food enters the digestive tract, but Ivan Pavlov, the scientist famous for ringing a bell and making dogs salivate, actually won a 1904 Nobel Prize for mapping what is known as the cephalic phase of digestion.
The cephalic phase, meaning related to the brain.
Yes. Pavlov proved that the mere thought, sight, or smell of food ties your central nervous system directly to your digestive organs. Thinking intensely about eating actually triggers gastric and pancreatic secretions before a single bite of food ever touches your lips.
Wait, really?
Really. Which means, by simply listening to this highly detailed deep dive about food and digestive chemistry for the last 15 minutes, your nervous system has almost certainly already started priming your stomach acid and pancreatic juices for a meal you haven't even eaten yet.
Wow. So just by conceptualizing the enzymes, you are literally triggering their release in real time.
Yeah.
The sandwich doesn't even have to exist yet for the machinery to spin up.
Pretty wild, right?
Mind blown. Thank you so much for joining us on this deep dive into your own inner machinery. The next time you grab lunch, I guarantee you will be thinking very differently about the microscopic choreography required to turn that sandwich into you.